Hi, I'm Miel Nicole Magtoto, and today I'm here to talk about 3D bioprinting of tissues and organs. In 3D bioprinting, layer by layer precise positioning of biological materials, biochemicals, and living cells with spatial control of the placement of the functional components are used to fabricate 3D structures. The first attempt in mimicking a working organ was done in 2002, where a miniature functional kidney was 3D bioprinted. There are many factors in choosing the material to be used in 3D bioprinting, such as printability, biocompatibility, degradation kinetics and byproducts, structural and mechanical properties, and material biomimicry. Aside from these factors, the complexity of the functions or how the human body works must also be considered. An example of one of the most complex systems in the human body is the circulatory or vascular system. Today, this is a challenge in bioprinting tissues with functional blood vessels. So in order to solve this problem, the team of Jennifer Lewis from Harvard University created a new bioprinting method which takes into account the complexity of the vascular system. Aside from the vasculature, they also proposed a new bioprinting method of multiple cell types and extracellular matrix or ECM. Gelatin methacrylate or GELMA was used as ECM due to its low cost, abundance, ease of processing, and biocompatibility. It is a denatured collagen that is modified with photopolymerizable methacrylate groups, allowing the matrix to be covalently cross-linked by UV light. For the embedded vasculature, Pluronic 127 was used because it can be easily printed and be removed under mild conditions. Also, in previous studies, it demonstrated effectiveness in printing synthetic microvascular networks. The same aqueous GELMA was used to create cell-laden inks dyed with fluorophores for 3D bioprinting. Prior studies have shown that cells adhere and migrate through the GELMA due to the presence of integrin binding motifs. The integrin binding bridges cell-to-cell or cell-to-ECM interactions, which regulates the cell cycle. This also allows the movement of the cell, which allows blood flow in the printed tissue constructs. The tissue construct was produced in a layer-wise build, sequenced by co-printing the ECM, vasculature, and cell-laden GELMA inks. Pluronic 127 contained either protein-expressing human neonatal dermal fibroblast or mouse fibroblast line. Another cell type, endothelial cells, were also added since these regulate the exchanges between the bloodstream and tissues. This process produced the engineered tissue constructs. After 2 days, using microscopy, the locations of the 3 cell types that were independently stained were identified. The human neonatal dermal fibroblast in green, mouse fibroblast in blue, and endothelial cells in red. Image shows that the cells created a network of tissues where they continued to develop. They tested the viability of the cells for a week, and it showed that its viability increased from 70% and 61% to 81% and 82% for the human neonatal dermal fibroblast and mouse fibroblast line respectively. The team of Lewis also filed a patent for their method of creating new vasculature, multiple cell types, and extracellular matrix. Their claims include printing tissues with complex networks of embedded vasculature with multiple cell types. In the Philippines, some research has been done regarding 3D bioprinting. Students from TIP have successfully replicated a trachea. It means that 3D bioprinting is possible in the Philippines. Since the study is about replicating blood vessels in the body, advancements in drug formulation or drug delivery methods can be improved. Or instead of in vitro studies, in vivo studies can be done.